Welcome everyone to section 2 of guided notes. This one, it has kind of a lot in it. It's talking about chromosomes and DNA replication. So here we go. So when we talk about DNA, we talked way back in mitosis, we talked about the S phase. And during the S phase of the cell cycle, DNA replicates and we copy the genetic information. And that's what this is going to be about. So in order for DNA to replicate, it needs to have time in order for this to happen for a bunch of different reasons. The cells need to make new cells is why we go through mitosis. Why do cells need a copy of DNA? Because they produce sex cells. That's meiosis. And the other reason why they're going to replicate or copy the genetic information, now this is not DNA replication, this is a little bit different, but the idea is to produce proteins. And we're going to talk about that replication or that production of proteins as we kind of go along. So when we talked about S phase of the cell cycle, that is when our chromosomes, our DNA is copied during S phase, right? So G1, the cell grow, S phase, the DNA is copied. And that's what we kind of just generically said. But now we're going to talk about the mechanism and how that actually happens. So in eukaryotic cells, remember you are a eukaryote, and the reason you are a eukaryotic cell is because you have membrane-bound organelles, you have a nucleus, is kind of what we often classified. So in order for us to kind of figure that out or to better understand that replication piece, it's important to figure out kind of some things, remember some things about the cells. So eukaryotes have a nucleus, and you are a eukaryote. And in that nucleus is the DNA. In the nucleus is the DNA, and that's important. And during mitosis, that nucleus dissolved, right? But it was housed inside the nucleus, and it's kind of protected there. So how does that DNA fit in there? Well, again, it's about 2 meters tall, so over 6 feet tall. If we were to take all the DNA in your body, in a single cell, and you were to put it end to end, it would be about 2 meters tall. That'd be taller than me and Mr. DeBoer. Mr. DeGrand called each of us, right? So if you were able to stretch this all out, it would be about 6 feet, over 6 feet tall in a single cell. And in order to kind of figure out how it can all fit in there, we were going to kind of talk about proteins and how it wraps around. So this DNA is twisted and wrapped around these proteins, and it's just coiled and coiled and coiled. So we have this long stringy thing which is coiled, and then those coils are all kind of then wrapped around. So right, again, here's our DNA molecules that are wrapped around proteins and then wrapped around and wrapped around and wrapped around and super coiled. So DNA is first twisted into what we call a double helix. DNA is a double helix. It's a double helix. You should know that DNA is a double helix. Watson and Crick kind of came up with that idea. And this helix is wrapped around small proteins called histones. There are proteins, DNA wraps around proteins. It's called a histone. And then what happens is those then coil, and then those coils coil, and then those coils, we get these ideas of chromosomes. So they're wrapped around these other proteins called nucleosomes. And then these are coiled up even more and more and more until they all are able to kind of condense and wrap up into the nucleus. So how does DNA replication start? Well, notice this picture. These are running in opposite directions. We call that anti-parallel. But Chargaff's rule, C's and G's, A's and T's, right? So that's the idea. So in order for DNA to be replicated, it first has to uncoil, and this is done small segments at a time. And then they split these hydrogen bonds. These are the hydrogen bonds right here. And what happens is we go through and we split this. So then what happens is they actually separate right along those nitrogenous bases. They are weak bonds, so they're easy to break, but they're strong enough to hold them together for most of the time, but they're easy to break. So then what all of this process is about enzymes, and enzymes are specialized proteins that go through and help reactions occur. So DNA is first split by a protein called helicase, which then breaks the DNA into something called a replication fork. So a protein kind of comes through and it unzips the zipper, right? If you kind of picture these being the teeth of a zipper, this is actually the zipper, what they call the zipper pull, and it unzips the zipper, breaks those hydrogen bonds. And the two strands of DNA are then built in opposite directions, starting at this replication fork. So in order for us to duplicate this, we have to keep in mind that we have these things called nucleotides. And there are two strands that are being created. So what we need to do is we need to make an exact copy. But instead of just making a complete copy, it's called a semi-conservative model. and each half of the original strand acts as a template or to make a complementary strand. So the idea is we have one side that we're going to split, and then using Chargaff's rule, we're going to build a complement. So there's an A and a T, and then the idea is if we were to split this right here, what are we going to bring in to match up that T? Well, it'd be an A, right? And if this gets split, what's going to come in to match up this A? Well, it's going to be a T, and you'd be like, well, they're going to be the same. Yeah, we'll have two strands that are the same when they put them all together. And that's kind of what this is showing. One of these runs in this direction and one runs in this direction. But the idea is another enzyme called DNA polymerase is another enzyme, helps chemical reaction. It builds, we keep calling them complementary strands because it's a complement. It's the other side of, it's not an exact brand new copy. It's the complement. It's the other side. And it adds one nucleotide at a time. So remember, these nucleotides consist of a phosphate, a sugar, and a nitrogenous base. And these are our four bases, A's, T's, C's, and G's. And as it keeps moving down the line, these DNA polymerases is doing some proofreading because we don't want errors if at all possible. Could cause mutations, could be good, could be bad, couldn't make no difference, but we try to avoid mutations or oopsies at all costs. Okay, so then at the very end, there is another enzyme that kind of goes through. So there's multiple replication forks that are going through to create a DNA strand. As these forks come together, they will create one complete strand when we're all done, and they connect them with this another enzyme called DNA ligase. And it makes sure that the sugar phosphate backbone is connected. So it kind of takes this piece and connects them in this piece and this piece and makes sure that they are connected. So we have two complete backbones. It's another enzyme. Lots of enzymes involved here. They call it a semi-conservative model because it's not an exact copy, but we use our original strand. We take half of it, and then here's the other half. So they kind of color-coded, so we kind of have this pink or purple color. So if you notice over here, this strand has one, and this strand has one of, right? It's half here, and it's half here. But if we bring in the complements to each side, what we end up with is two complete strands. So the new way DNA is replicated is called semi-conservative model, and this is because the new strand serves as a complement, or each new strand of DNA has one half from the original strand. It's not a complete new set. It's half of conservative. We conserve, we keep. Okay, so we somewhat keep our original strand. Okay, so that is the idea with DNA replication. Lots of enzymes, Chargaff's rule, complements get matched up, semi-conservative model as we kind of work through this. So that's section 2 on DNA replication. Let us know if you have any questions.